This summit can help ensure that artificial intelligence charts a course that benefits humanity and bolsters our shared values.
Welcome to AI for Good, the leading action-oriented, global and inclusive United Nations platform on AI. Organized by ITU, in partnership with 40 UN sister organizations, and co-convened with Switzerland. The goal of AI for Good is to identify practical applications of AI to advance the United Nations Sustainable Development Goals and scale those solutions for global impact. In today's session, we're counting on you to use the live video wall feature to ask questions and post comments to help create an engaging discussion. We encourage you to stay until the end to chat, connect, ask questions, and network with our distinguished panelists and world-class AI experts in the neural network. It is now time to kick off the session and welcome our first speaker. The floor is yours.
Good morning, good afternoon, and good evening, everyone. I love that new intro highlighting the AI for Good Summit in person in Geneva, July 6th and 7th. So everyone mark down these dates in your calendar. My name is Matthias Greschel. I'm a resident physician in internal medicine at Charité Hospital in Berlin, Germany, and a research fellow in biomedical and informatics at Harvard Medical School. And I'm really delighted to welcome George Ripsack today, giving a talk about how to draw reproducible conclusions from observational clinical data. Dr. Ripsack is Vivian Beaumont Allen Professor and Chair of Columbia University's Department of Biomedical Informatics, and he's also the Director of Medical Informatics Services for New York Presbyterian Hospital. He's not only a data scientist, but also a board-certified internist with degrees in chemistry, medicine, and biostatistics. Dr. Ripsack's research focuses on the clinical information that is stored in the electronic health records and also the development of next-generation health records. He is leading the Observational Health Data Sciences and Informatics Coordinating Center at Columbia, which is a huge international network with thousands of collaborators holding records of almost 1 billion patients. That is very impressive. George, please come online if you're there. Dear audience, please ask away. You'll all be following the talk on the neural network, so you can post your questions in the chat and I can ask these to George along the talk. And we'll also be online later to chat with all of you. And George, please take it away from here.
Thank you very much, my friends. Let me just share my screen. Was that successful?
That looks successful. Perfect.
Okay, thank you. All right. First, let me say I have nothing to disclose financially. I'm going to be talking about observational research. I'll be talking about big data and data science a lot. Some of it is AI, some components are more AI-oriented, some are more traditional data science, but I think it fits together. And as time goes on, we're increasingly relying on AI. So what do I mean by observational research? Observational research is where subjects are observed in their natural setting. It's also known as real-world evidence, as opposed to something experimental like a randomized clinical trial where you engage people in the trial and measure them along the way. Here, we're just looking back at people, look back at their insurance-based administrative claims data, look back at their doctor-based electronic health records, and so on. If you just look at the US as an example, it's $4 trillion. That's a lot of experience that we get out of that, and we should be learning from that $4 trillion worth of effort. In other words, in the US, a lot of health research is funded by the National Institutes of Health. They can't afford to duplicate this. We have to learn from everything that we do. Now, if you look, do we need more evidence? Well, this chart shows on the left side, all drugs around the world. And on the top is all possible things that can go right or wrong by taking a drug, all possible outcomes. And what's shown in red dots is where we have evidence in the literature about whether a drug causes a side effect or a good effect. And as you see, it's what we want is a red rectangle that is filled in evidence. But what we see here is a mostly white rectangle, and we cheated. Those dots are 10 times the size that they really are, because if I did the real one, you would just see white and wouldn't understand what I was saying. So we need to fill in a lot of this evidence. When a doctor prescribes a drug, the doctor should know what are the side effects of that drug and not just a smidgen of what is needed. We need large databases because you might start with a 2 million patient database, but by the time you pick a disease you're interested in, and a drug that you're interested in, a side effect you're interested in, often we'll get down to, say, 25 patients left in the study who follow that, who fulfill all the eligibility criteria. By increasing that by a factor of 1,000, I can start doing real studies. But there's a catch. When you do retrospective research, all kinds of biases can come in. Here's an example, 2 top journals, JAMA and BMJ, answering the same question, do a drug called bisphosphonate cause esophageal cancer? These are based on the same database and published a month apart, and one says yes and one says no. So the public and the lay press read this, and they're not sure what to do with these contradictions that they see. And then with COVID, there was a flourish of research occurred, but there were retracted papers when there was concern, for example, about the veracity of the data and the analyses that were done. The end result is editorials like this, which the title is The Magic of Randomization versus the Myth of Real-World Evidence, saying the only trustworthy thing is an experiment, a randomized clinical trial. And assembling these big databases is prone to problems. My observation is that these editorials don't get it quite right. We're not comparing observational research to randomized trials. If we can do a randomized trial, that's great. But the problem is that we can't do all the randomized trials we'd like to do. They may not be ethical. We can't force people to smoke or not smoke cigarettes. And we don't have the money to do all the experiments we would like. So what happens instead is we rely on expert opinion. So we take a guess, basically. And observational research, I believe, if done correctly, is better than just taking a guess. And people have come up with frameworks. This is by Patrick Ryan of J&J in Columbia, which is a framework for judging whether evidence is reliable. And there's words like repeatable, reproducible, replicable, and different components on which we would test whether if we change researchers, change databases, change analyses, do we get the same answer? The important thing for this slide right now, though, is that we can answer that question, we can address this framework if we have a community of researchers working together, a community of populations on which we can test it, and so on. So what we've done is to create such a community called the Observational Health Data Sciences and Informatics Community, OHDSI, which we pronounce as OHDSI. OHDSI's mission is to improve health by empowering a community to collaboratively generate the evidence that promotes better health decisions and better care. It's multi-stakeholder, international, and Columbia University, where I work, is the coordinating center. Here are our current participants in the network. You can see we have over 3,000 collaborators as judged by, we use the Microsoft Teams environment to collaborate. We have over 3,000 people defined from 80 countries. We were founded in 2013, so going on 10 years ago, we've published almost 500 papers, some of them very impactful. I'll show you some of the results towards the end. How does OHDSI work? Well, it's an open science effort, which means in part we have all our software is open source, but beyond that, we share everything. And I'm going to come back to this point. Openness is critically important for reliability. So how do you get all these people to participate? Well, we have a federated database, which means everyone keeps their data local. You take your database and you transform it to a common data model, keeping your database local. And then we distribute the query around the world. It's a voluntary network, so people run what queries they want and send the results back centrally. We then analyze the results collaboratively with everyone who participated and publish the papers on it, again, collaboratively. So we're drawing data from all around the world. So the first problem is that there are different languages and different coding schemes. And as you know, as you try to bring an appliance around the world, it doesn't fit and we don't want to rewire everything. So we come up with a common adapter to make it look the same, and that's the OHDSI OMOP Common Data Model. This is the set of tables in our data model. It's a deep information model. As you can see, we try to keep it simple. So you can see tables that have names like person, visit, drug, procedure, device, measurement is where the lab tests are stored, and so on. So we've created this database which people put this database format that people put their data into. Now, if you're coming from many different countries, you're going to have different vocabularies. And what we do is we translate to a standard core set. So you may start with read codes or ICD-9 CM or ICD-10 non-CM or ICD-10 CM and so on. We convert all those diagnosis codes to something called SNOMED CT. Medications in ARCS norm or ARCS norm extension, and lab tests in LOINC, and a couple of others. We have 10 million concepts in our vocabulary and 80,000 relationships among those concepts. Now, anyone who's done work in standards and converted data to a data model, has some experience, knows that if you give 3 people the same data model and the same data, they'll come up with 3 different ways of translating it. For example, if we're trying to encode in our database the fact that someone passed away, that someone died, what do you put as the diagnosis? Is it that their heart stopped? Is it that they had pneumonia in the last couple of days, or that they had a fatal cancer that then precipitated the pneumonia, which then led to their heart stopping? So a lot of what we do is about standardized conventions to give people advice on how to encode the records so that we all encode them in the same way. And then we give you tools, which I won't go through in detail, to convert the data so that can be used around the world to speed the process. And one of the most important things, if we want to have reliable data, is to have high data quality. So we have a set of tools that verify the data quality. People who have participated in OHDSI have sometimes told us the most valuable thing they got was being forced to convert their data to the OHDSI data model and running the data quality tools, because they learned there were problems with their source systems that they needed to go back and fix that they never knew about until they converted to the OHDSI data model. When we do this, we've managed to collect, 453 data sources contain data that have been converted to the OHDSI Common Data Model. When we add it up, and it's shown in orange on the map here, countries that have participated in that way, 928 million unique patients have had their records converted to the OHDSI OMOP Common Data Model. That's 12% of the world's population done without paying people. So they've done it voluntarily. Now, I have to tell you that when we do a study, we don't necessarily have 928 million patients worth of data because it's a voluntary network. Usually it's a subset of that. It's in the hundreds of millions, but not quite at the 900 million. Now we have this database. We have 10 million terms in this database. If someone wants to do a study, we need to help them. So we have tools to help them work through those 10 million terms to find the ones that represent the disease they're interested in, the drugs they're interested in. We have tools to help put those things together to create queries, to create concepts that you do research on. And then we have tools that help you put your study together and put in the statistical analysis, say, a survival analysis to find out whether one drug or another drug has worse side effects. And then we visualize those results. OHDSI divides so OHDSI is an evidence generation initiative. It has a database, it has a data model, it has a vocabulary, but the primary goal is generating evidence, reliable evidence. There are 3 types of evidence. The first is clinical characterization. So that means just counting how often things happen, like how many people have diabetes and what drugs do people take. That's the easiest one in the sense that I don't have to draw strong conclusions. The second one is population level estimation. That's where we get to causality. This one is harder, and I'll be talking about this. This is where we test hypotheses like, does metformin cause lactic acidosis? or does it cause more lactic acidosis than glyburide? And the third is patient level prediction, where we say for individual patients, whether they are more or less, say, at risk to have a side effect. Let me just do a slight sidebar on phenotyping. So we have these raw data stored in our database, and we brag how big our databases are, right? And so Columbia's database is 6.8 million patients, which we use for OHDSI research. But when someone tells you how big their database is, a lot of those databases, a lot of those patients are like a single point, like on the lower right there. So one problem with electronic health record databases is that data are sampled mostly when someone's sick. We're missing all the days that they're healthy. We're not getting those measurements. Furthermore, the electronic health records are noisy. Some things are as low as 50% accurate, like smoking history. And that second line, 36-year-old man, 27-year-old woman, is pulled from a real paragraph in the electronic health record. And it's complex. If we just look at time, what we want is when the thing happened to the patient, which is, say, when a lab specimen was drawn. We often don't have that. We have the specimen received in the lab, the test was performed, or when it was received by the patient, or when the clinician wrote in the note. So we don't always get the time quite right. And furthermore, healthcare is a big feed-forward network, and what you do to the patient affects when you measure the patient next. That causes biases. But the good news is that doctors use the electronic health record successfully. They have a good feel for what's going on. So when we use the database, we need to mimic the doctor's reasoning and deconvolve the truth. We split the process in two with a process we called phenotyping, which is a reuse of a term from biology. We go from the raw data to a phenotype, and that phenotype is like, say, does someone have diabetes? And then from there, we use that in our analyses. So if you want to know if someone has diabetes, you don't just look up a diagnosis in the record. You might look at their medications and their glucose, other diabetes tests, and mentions and notes. And producing those things can take months to define and test. So OHDSI has created a phenotyping pipeline that goes across the network and exploits what we've learned to help to guide the phenotype author in how to create these phenotypes, what codes to use, how to assemble that into a rational thing, and how to test it automatically so that you don't have to do manual review in the chart. Again, this is just a short section, so I'm not going to go into detail on this, and I'll move on to the next part. Let me right now give you an example. It was that first type of evidence that I called characterization, that is tallying things. And what we did is we looked at treatment pathways, and this was our first large study in OHDSI. If you consider, like, how do a doctor and patient decide to take a drug? Well, there are many influential things. There are guidelines shown in the middle of the figure, but there are many other factors coming into it. And we set about to study, well, what drugs do people actually take as opposed to what is recommended in the guidelines? We did a study across 5 countries with about 240 million patient records. We published it in the Proceedings of the National Academy of Sciences in the US. Here's an example of one of the outcomes. So this is a starburst plot. The inner circle shows you what drugs they took first. The next circle out shows you what drugs they took second, third, and so on. This is for diabetes, type 2 diabetes mellitus. And we see from the inner circle, it's mostly that yellow-green color. 75% of the time, patients take a drug called metformin, which is good because that's what the guidelines recommend, so that helps us. You can see that 30% of the time, metformin is the only drug they take in a 30-day period, means that 45% of the time, they actually also take a second drug, so metformin's not completely working. We then did this across these 5 countries, and we had some interesting results. If you look at the first column, now there were 12 databases. I'm only showing 3 of them in the first column. The first is the US, the second is UK, the third one is Japan. And you'll notice, now we also studied Hong Kong, we studied South Korea. Everything looked like the US and UK, where they mostly use metformin, which is that big yellow-green circle in the middle. Only Japan looked different, where they only use metformin a third of the time. a quarter of the time total. We talked with a Japanese diabetologist, and they explained that the Japanese doctors believe that Japanese patients get insulin resistance less of the time. Genetically, metformin is intended for those patients who get resistance, so they tend not to use the drug. Even though patients nearby them in Asia Pacific still use metformin. So that was an interesting surprise finding. The second column is how we treat hypertension. There's less agreement there, but still some. The third column, there's almost no agreement, and that's depression. And if you know about depression, we don't really know that well how to treat depression, how to match a drug to a patient, and that's why we see the least agreement. One interesting thing was 25% of hypertension patients took a sequence of drugs in 3 years that was shared by no one in our 240 million patient database. So patients, 25% of patients are taking a sequence of drugs, and they're unique in the world. I mean, unique in this 240 million. So that was interesting that doctors are doing very different things and not necessarily following guidelines. Our conclusion from this study was that it's feasible to encode data in a common data model. People are willing to volunteer to be part of studies, and we're able to address privacy concerns and research regulation across many countries by federating the database. Now I'm going to come to an example where we test hypotheses and do causal inference, what we call population level estimation. So first, remember I said that the literature has problems. I'm going to show you a study of the literature that we did. And I'm showing you this figure on the x-axis. On the horizontal axis is the hazard ratio. That's like the study's outcome. And a 1 means the drug has no effect. And on the vertical axis is the error in that estimate, the estimated variance, or the standard error. Now, if you look to interpret the diagram, the thing on the far right in blue means that the drug is harmful. The thing on the far left in tan or green or what color you see is that the drug is beneficial. And the purple on the top means the drug was not a statistically significant result. We parsed 30,000 studies in the literature, and we found that 85% of them show the drug has a true effect. In other words, only 15% of drugs didn't have a side effect or didn't have a particular side effect. So that's not feasible. And you'll notice that the figure is kind of missing a triangle there. It's kind of empty there in that purple spot. This is something called publication bias. What it means is that if you have a study and it doesn't show that the drug is harmful or beneficial, you don't publish your paper, or the editor doesn't publish your paper, or the reviewers are not excited about the paper. So what happens is we do a lot of studies, and only the studies that show positive results get published in the literature. The problem with that is then we don't know the denominator of negative studies to help us interpret the positive ones. We also see this other effect where it's enhanced along the border. Authors know that if they get a study result which is not statistically significant, they're not going to get credit for it because they won't be able to publish it. Sometimes what happens, and usually it's unconscious, they try different things, different analyses, different variables. Suddenly they get a statistically significant effect. The p-value goes from 0.051 to 0.049. And then suddenly their paper becomes publishable. That's called p-value hacking, and that's treating, and that's one of the reasons for unreliability of the literature. People say, well, 85% of them are positive because we're only studying the things we think we're going to be positive anyway. Why would we study something negative? And we're just guessing, right? Well, this is a cross-section of that figure. And what we see there, the red line, is what happens if you're perfect at guessing which studies are going to be positive. And the blue line with the cliffs show you what happens when you have publication. That's what we see. You can only get those cliffs if you have external bias. It's not because we're guessing right. So individuals may produce good research, but in aggregate, the medical observational research system, that is the literature, is basically a big data dredging machine. We're picking out only the things that are positive, and they may be chance. So OHDSI came up with a set of principles to try to avoid this. The 10 principles here, I won't go through in detail. I'll just summarize them on the next slide visually. You can summarize as saying that on the right is openness, on the left is verification. So openness means I need to tell you ahead of time what study I'm going to do. That way, when I don't publish it, you still know that I tried to do the study. I need to make all my software freely available to you so you can duplicate my study and try to replicate it. When I get results, I need to put it on the, and need to put diagnostics on the internet and then my results on the internet so you can check and make sure that I didn't cheat somehow, that I didn't change the analyses to try to get a positive answer that I thought I could publish. On the left, diagnostics, making sure that my data are high quality, that my analyses go through correctly, and that I don't find any contradictions in there. And I do that before I look at the study results and try to publish my paper. And we believe if you follow these simple steps, we can get a more reliable literature. So here's some going a little bit deeper in some of those things, how OHDSI addresses reproducibility. So I'm going to go into more detail. So I'm going to skip over the slide and go a little bit deeper. One of the big problems in retrospective research is something called confounding, which is where you're studying one thing, but you're getting the effect of something else. So let's say I want to study whether butane gas causes lung cancer. If I do a simple study in the population and say, who has butane gas near them and who gets lung cancer, I'm going to find a positive association and say, hey, maybe butane gas causes lung cancer. But if you look more closely, you might say, well, the only people who are carrying, most people who are carrying butane gas with them are carrying cigarette lighters, and it's because they smoke, and it's actually smoking that causes the lung cancer, not the butane gas. So that's called confounding. I'm trying to study the gas, but I'm seeing the effect of the smoking. So if I'm trying to study a drug, I want to see the effect of that drug, not something else. So we try to correct for confounding. One of the main ways that we correct for confounding is something called propensity score adjustment. So what we do here is we say, I'm comparing two groups of people, and I want to make sure that they're similar in all ways, except one took a drug I'm interested in, one didn't. Or put another way, taking the previous example, I want to compare two groups of people, some who have butane gas near them, some who don't have butane gas near them, but they smoke in equal percentages on both sides, that they're balanced, so that we don't see the effect of smoking, we only see the effect of the butane gas. For example, we can compare people who smoke who have matches versus people who carry cigarette lighters, say. So the propensity score, as I said, balances things, but in order to carry it out, we need to select the confounders. That is, we need to identify the things we're worried about, like smoking in that example. Traditionally, it's said that you have to manually select the confounders and you have to get them right. You can't adjust for non-confounders and you can't miss any. So I looked at 5 studies of hypertension and said, well, if they're always getting it right, let me see what confounders 5 different papers used. And you can see from this list that they, in fact, picked 5 different sets of confounders. So at most, one of them got it right, but at least 4, and probably all 5, don't have the list exactly right. A second approach is empirical selection, where you try to learn it from the data. And I'll come back to that. What we do is something else. OHDSI, when OHDSI looks for solutions, it tends to go large scale and systematic. So what we do is we actually balance on every covariate we can find, every variable in the electronic health record. So 10,000 to 100,000 different variables, we balance on all of them. The problem on that is that it can create problems if you balance on things you're not supposed to. And I won't go into detail on this talk, but there are things called mediators and simple colliders and instruments and M-bias. And this procedure that we use tries to eliminate all those kinds of variables by only looking at variables that happened before the intervention, by using domain knowledge, by using diagnostics, and some other things I'll show later. So OHDSI uses this large-scale approach. And it seems to work. So shown here, so remember, I'm comparing 2 groups, someone who took a drug, someone who didn't take a drug. And what I want is for those 2 groups to be balanced in all the confounders, so that if there's 10% of people are smoking in this group, then I want 10% of people smoking in the other group. This figure is a plot of 60,000 variables. Each of them is a purple dot. And what I want is to see how balanced the two groups are. On the x-axis is before I did my propensity score. And I want all the things to differ by no more than 0.1, which is a threshold that was set up outside of us by Graham. And as you can see, before the balancing, the groups differed by more than 0.1. But after we did our propensity score adjustment, everything is under 0.1. In fact, everything is under 0.05, and almost everything is under 0.025. So we achieved excellent balance. And we further, we verify that when we achieve that balance, we don't mess up the study in other ways by reducing our generalizability, but I won't go into detail on that. Now, the problem comes up is this. How do I know there's not a confounder, some factor that's going to mess up my experiment that's not measured? What if I'm missing a variable? Like my electronic health record may have 60,000 variables, but what if I'm missing the one important one? So we did a study on hypertension, and there was an important variable that was missing from most of our databases. It's your measured blood pressure before you took the drugs. That's called baseline blood pressure. But fortunately, we had one database or two databases that had the baseline blood pressure. So what we did is we did our propensity score adjustment with and without blood pressure on that database or those databases. Well, I'm going to use one database as an example. And what we found was blood pressure was correlated with those other 60,000 variables, so that it didn't matter, the results on the right-hand side, it didn't matter whether we included blood pressure, because when I balanced on the other 60,000 things, blood pressure was correlated with it and ended up being balanced too. So then we say, well, how far can we go with this? We did a study where we deleted every diagnosis from the record. No diagnoses. So I don't know what diseases the person had, but I balanced on the medications. And what I showed was that when I balanced just on the medications, every single diagnosis ended up being balanced in that study. In other words, medications and diagnoses are sufficiently correlated that balancing on all of one ended up balancing the other ones. And then we used it in practice and showed that, that doing this was better than manually selecting the confounders. I won't, again, for interest of time, I won't go into too much detail. And then we published further studies showing that it's better than empirical selection or manual selection. Another thing we do is diagnostic, and we call them negative controls. A negative control is something where it's just like the study I want to do, but I flip something, I change something to where I know the answer is no. So if I'm studying aspirin, and does aspirin cause an ingrown nail? No. I know that an aspirin doesn't, at least in the short term, cause an ingrown nail. It may be used in the setting of an ingrown nail to address pain, although not so much these days, but it's not going to cause an ingrown nail. And OHDSI uses 50 to 100 negative controls. So what we do is we repeat the experiment that we want to do, is this drug working or not, with 100 other experiments where we know the drug doesn't work. And we see how often does it tell us that, does it think it works? The number where it gets it wrong is supposed to be 5%. That's the definition of a p-value in statistics. So we expect when there's no effect, we should see a false positive effect that we shouldn't see 5% of the time. If it's more than 5%, we have a problem. Here we did it, we got 16%. So we recalibrated, we widened our confidence intervals so that we brought the rate down to 4%. The important point on this slide is that we have diagnostics, that independent diagnostics that tell us, is our study reliable? And that's really the point I want to make there. Another thing we do is we look at many databases, many locations, many practice types, and look for consistency. And we're open, as I mentioned earlier. We publish all our hypotheses, code, parameters, and runs. And we tend to study a lot of things at once, and that gives me the operating characteristics of the analysis. In other words, if I do a study and I show that every drug causes blindness, well, I know that's not true, so then I'd be worried that there's something wrong with my study design. When we use these methods, When I plot the same diagram, so in the lower left, the little diagram shows the literature. When we do it, we show that diagram in the middle, which has a cloud. In other words, we don't drop all our positive results. We don't do p-value hacking and so on. We find 11% are positive when calibrated, which means 5% are false positive, and 6% are real side effects of drugs, which is a reasonable number just based on previous knowledge. Now, people say you're doing all these studies. Isn't that data dredging? I mean, you said you didn't want to do data dredging. Data dredging is when you throw stuff out and don't tell people. If you run a lot of studies, but you're honest about it and show them what you did, then they can make decisions based on that. If you run a lot of studies and throw most of it away, as the literature does, that's what data dredging is. Okay, now let's use it on a real problem that matters. So hypertension is very common all around the world. Here's an example of the US guideline. The European guideline came out about a year later. This guideline had 57 drugs. Half of them were first line, which means this is what you should use first, and half of them were second line. For the most part, they say, here's 29 drugs. We're not sure which one you should give. Use your own judgment. Guidelines are supposed to be evidence-based, that is based on randomized trials, experiments, but there were only 40 randomized trials that were relevant. This diagram there are little circles, little dots around a circle. Each dot is one of those drugs I mentioned earlier. Each arc shows you where we had a randomized trial that gives you evidence. A lot of drugs, a lot of those dots don't have any arcs to them. And then even the ones that have arcs only have arcs to a couple of other drugs. Yet we're making decisions on which are the better drugs. So most of the decisions in the guideline are actually expert opinion. We set out to fill this circle. So we did a lot of experiments comparing all drugs against each other. And since in hypertension, you often take two drugs, we compared all pairs of drugs against all other pairs of drugs, which is 164 million hypotheses, but most people don't take most combinations. So we ended up with about 500,000 hypotheses. And we designed it to mimic the way a randomized trial would work. We had 58 outcomes of interest, that is, things we wanted to happen and things we didn't want to happen, side effects. And we had those negative controls, like, for example, hypertension drugs don't normally cause an abnormal pupil or an absence of a breast. We used 9 databases, some electronic health records, some administrative claims from different countries. Here's an example of the results. In this diagram, purple means the drug on the bottom is better, green means the drug on the left side is better. Randomized trials shown on the left didn't differentiate the drugs, but on the right, our study, which is called Legend, has some purple and green. What it shows is CBBs, that is cardioselective beta blockers, seem to be worse than the other drugs on average. And that is, in fact, turns out to be true, and the guideline duplicates that. We had an unexpected finding, which is that diuretics are better than ACE inhibitors, and we're still looking into that. If I look at it in more detail, instead of drug classes, drug by drug, what this figure basically shows is that the US guideline is pretty good, according to our evidence. On the upper left, you see that it's mostly gray or tan, and the few purple and green are drugs that didn't pass our diagnostics. On the upper right is green, on the lower left is purple. That means that the first line drugs are better than the second line drugs. On the lower right, there's a mixture because the second line drugs, some are better than each other. Now, I'm going to go into a little bit more detail about a particular hypothesis, chlorthalidone, which is a diuretic, one drug, and compare it to other drugs, hydrochlorothiazide. Hydrochlorothiazide is the most used in the world. And they used to teach us when I went to med school that it's limited duration, 20 hours a day instead of 24 hours a day, help the kidney recover. Chlorthalidone is a drug that's slightly newer, it's longer lasting. And recently, the academic field feels that people should switch to chlorthalidone as being more effective. But everyone uses hydrochlorothiazide, so they're trying to push the clinical field over. As evidence, they use something called an indirect meta-analysis and some old observational studies that I believe are not that reliable. But a recent huge and very nicely done observational study in Canada by Dalla showed that hydrochlorothiazide is just as good as chlorthalidone and has fewer side effects, and that's what we should use. So we've repeated the study. We published it in JAMA Internal Medicine. We passed all our diagnostics. We showed that the two drugs are equally effective, but that chlorthalidone, which is the one favored, in the guideline and by the academic community, has many more side effects. So the author of the guideline and the former head of the US Center for Disease Control sent in a letter saying basically ignore our study, and they didn't even mention Dalla's study. They said there's moderately strong evidence that chlorthalidone is better, and you should just ignore this for now until a large randomized trial comes out. So that large randomized trial came out, was published in the New England Journal of Medicine, and showed the same results as us, that the two drugs were equally effective, and the one of them had worse, at least what the main side effect, hypokalemia, was indeed worse. The editorial in the New England Journal of Medicine said, results suggest that chlorthalidone therapy remains a good choice, and they don't mention anything about hydrochlorothiazide. So here we have our study, Dalla's study, and now a randomized trial all say the same thing, that they're equally effective and hydrochlorothiazide is better because it has fewer side effects. Yet the editorial that accompanied the randomized trial says this is evidence that chlorthalidone works. So it just shows you that when you have a strong bias towards something, that it's hard to let go of it. And that we'll see what the new guideline says. I suspect, despite the randomized trial with 13,000 people in our study and another, will still favor the old drug. So when we come to reliability of evidence, there's also the reliability of getting it into practice. And it takes time to convince people. On the other hand, if a field is already convinced of something, so we did another study that said that ACE inhibitors are better, that one drug is better than another drug. ACE inhibitors are better than ARBs are better than ACE inhibitors. And for that one, there was an editorial published in Circulation, a very important cardiovascular journal, which says, this study that we produced in Odyssey came out, it's time to change the guidelines and say that ARBs work. So if you have a prior that a drug works, you'll accept the evidence. If you don't, then you may not. So we said, well, how else can we prove our studies are working? We compared the studies we did to 30 of the randomized trials where they overlapped. And we got the same answer in 28 out of 30. The definition of a p-value is that you should get it wrong 5% of the time. We got it wrong only 6% of the time. So we're matching what's expected by chance. And then to show you that earlier diagram, we filled in that diagram with evidence and published a number of papers. And now, just as on the last slide, just a little bit about COVID. One of the studies we published was on hydroxychloroquine. If you remember, the world was using hydroxychloroquine early on based on relatively flimsy evidence because we had no alternative to treat COVID-19. A number of studies showed eventually that it was not effective. On the other side of it, we needed to show that it was somewhat dangerous. It's actually a very safe drug, but when you try to give it to 100 million people, then the side effects stand out, especially if you pair it with an antibiotic. And so Odyssey's study on the safety or lack of safety, a lack of complete safety, because it is very safe, but lack of complete safety of hydroxychloroquine was one of two studies that the European Medicines Agency used in determining that people should stop using hydroxychloroquine. There was another case where they were worried that blood pressure drugs might make your COVID-19 symptoms worse. So some studies came out, one of them from Odyssey. What I highlight here is that the European Medicines Agency highlighted the Odyssey study for its reproducibility as an exemplar of how to do observational research, and that's shown in the red underline. And lastly, March of 2021, 13 European countries suspended use of the AstraZeneca COVID-19 vaccine because fear of clotting. We had been working in Odyssey for 4 months on calculating baseline rates of adverse events more accurately. They asked us to calculate the rates for them for this analysis. And a few days later, on March 18th, they determined that AstraZeneca vaccine benefits on COVID-19 far outweighed the risks of clotting. based on our rates, which were then when we had time, we published it in BMJ, shown below. So that means that basically 700 million people had access to the AstraZeneca vaccine based on the analysis we were able to do for the EMA. And we published a number of studies there. We've been collaborating with a lot of agencies, the US Food and Drug Administration, the All of Us Research Program in the US, the eMERGE Network, National COVID Cohort Collaborative, the European Health Data and Evidence Network, or EDEN, a 30 million euro initiative to bring the databases of Europe together, followed by the Darwin Initiative shown on the bottom. And other countries, for example, South Korea adopted our data model as their national standard. So there's a lot going on. So in summary, current observational research that's published in the literature is in fact suspect. It's reasonable that people are suspicious, but we believe that large-scale observational research that's open and verified, as I said earlier, appears to be possible and more reliable than the current approach. Shown on the lower right is the funding that we've had for the initiative, but that's a minor part of the huge voluntary network. Here are the groups that I collaborate with most, but as I mentioned, OHDSI is actually a much bigger network of 3,000 people, and hundreds of them are very active, like many times per week kind of active. And that's it. I will stop there.
Thank you, George. That was fantastic. I encourage the audience to put in your questions, and we'll see them here during the discussion and can talk about them. Please do come forward with all of your questions. George, I want to start by asking, so if you're in the hospital I currently work in, if we wanted to join OHDSI, it seems like a daunting task to put in data that is hosted in different data silos where radiology and micro and so forth have different data types and different data silos. How does the funding work? Where do you get the personnel from to put it into the data structures outlined by you? Yeah.
So there's two sides to that. First is putting your data together. And then second is, specifically putting it in our OMOP OHDSI format. I think that the tide has turned and the incentive to put it all in one database is very strong now. And that hospital, say you take a hospital or a medical center, the financial incentives to do that are strong, both from, in the US, there's the way we do our billing creates an incentive. But even without that, if you look at AI's use in health, as we try to I didn't talk about prediction so much here, but as you have a predictive model and you want to validate it on your cohort, you need a database that contains all those data. If you want to create predictive models, similarly, you need to do that. Actually, not shown in my slides is a data consult service, which is a service where we actually run these studies. We can now, because of the nature of the OHDSI tools, we can run an observational research study in a day or two now, instead of saying 6 months. Because we don't have to, we have like an automated chart review instead of manual chart review and things like that. It may not be perfect, but it allows us to do that. So we opened up a data consult service where we go on rounds and see if there are questions that are not answered by the literature or otherwise. And then we could do a study for them in that day or two and answer it in time to hopefully affect the patient for the better. And our study, we believe that we do in a day or two is better than most of the stuff that you would have done in the old days. So once things like this become available, predictive models, if data consult becomes available, you'll need to have an aggregated database. So I think there'd be a strong incentive to bring together the pharmacy, radiology, clinical lab, and the notes. The OMOP common data model, I think, is a data model that's been used by many programs in North America, Europe, and APAC. And we're starting to get inroads in Africa and South America. So it's beginning to become more and more, it's one, and then it comes with a set of open source tools that you too can add to. So that's the incentive there, just become part of the community. So the funding has not been in Europe, the EMA decided, EMA and the big European funder whose name I'm blanking on right now, first them and then EMA decided to put money into this because of the benefits. But most of OHDSI has been voluntary, where each group decided it was worth their while.
Yeah. And so Leila is wondering, how can you access these data? Is it purely based on, can you can you sort of put in a request for data use, or is it those who collaborate and contribute? Is there a coordinated effort?
Yeah, exactly. So it's a voluntary network. They run it, so you have to be able to create a package, an OHDSI package, which is, it uses R programming language. It doesn't have to be an R. R is just a shell, and you could put something else in there. We use C++ and other things for speed. But that R package gets distributed on GitHub. They've downloaded it. So how do you get people to run your package? Well, one, you have to learn the OHDSI Common Data Model in our OHDSI stack so that you can build a programming language that goes on top of it. And by becoming, it's not a requirement, but by becoming part of the community, you get to know everybody. So when you say, I'm running the study, will anyone run it? Someone will run it for you. If you happen to have a database, you can say, I'll run your study if you run my study. But you don't have to have a database. You can just participate. Usually, like if you're donating your time towards the effort, people will be willing to donate their time back. So there is no request form. I mean, there is a process we have for advertising that a study is available, and that's our Tuesday call, which is at, I guess, 5 p.m. Greenwich on Tuesday every week. No, wait, no, 11. It's at 11. I'm sorry, I got that wrong. 4 p.m. Greenwich. It's at 11 a.m. Eastern.
Okay. All right. Okay, that's good to know. George, how do you account for the fact if using like sort of data collected in hospitals that you don't have patients who do not have insurance, who do not seek healthcare, and so forth? That kind of bias that is probably hard to address.
So we have a work group that is working on health equity. So there's an OHDSI health equity work group that concerns itself with this. I mean, we try to triangulate that people do show up in different systems. So there's the public, like in the US, I'm more familiar with, and there's the public, the kind of the systems of public hospitals and private hospitals. And then there's the insurance data versus the electronic health record data. Still, there are people staying home and not getting treated, and we miss them. And so then you can try to triangulate, then you could do individual experiments like social work services things where you discover things indirectly. You don't have the medical details, but someone had a problem. So I guess I'll just say that it's a recognized problem. We agree, and we have a work group that works on that.
Okay, yeah. So Fabian Rudolph is asking, he's referring to those not donut plots, the drug charts.
Oh, the starburst?
The starburst, exactly, where he was wondering where those where the drugs displaced used for all conditions or only to treat the mentioned diagnoses.
Well, that's a good point, because how do I know metformin wasn't used in some other way? They were all patients who had the disease and took the drug, but I can't guarantee that the indication was actually the disease. I think an overwhelming majority of people with diabetes taking metformin, it's something to do with controlling their glucose and not something else. But for hypertension, because you have hypertension, there's a lot of drugs there that could be for heart failure or something. I agree with that. Depression is probably right. Maybe hypertension is the one that's most at risk for that bias.
Yeah, and there's, also I'm just thinking of SGLT2 inhibitors, which are now diabetes and cardiac failure and kidney failure. So there's, that makes sense. So as I'm very interested in infectious disease and TB, in particular tuberculosis, do you have, just out of interest, how do you have lots of cases for these types of diseases, which is like for the United States and most of Western Europe, mostly low incidence countries?
Yeah, we're just beginning to have databases showing up in parts of Asia, Africa, and South America where there may be more prevalent. Remember, some of our databases go back to 1979. So in fact, in New York City, there was a lot of tuberculosis in the 1990s. So our database is a place where you can do tuberculosis research, not with modern drugs, but with the drugs that were available in the 1990s. But yes, and that's why we've tried to focus on bringing in groups from around the world. we don't have one huge funding source to help us. So we try to get grants from foundations to help us out, but largely it's been groups having to find ways themselves into the network. So we've done pretty well. I mean, but we recognize the biases there. You know, you can also look at other types of data we don't have, like we don't have, you know, Fitbit
imaging. Yeah.
We have imaging. We have the reports.
Yeah.
For places that can parse them. So like we have the reports, we don't put the images. It's not that it's not possible. It's just that, you know, most places it was a struggle to, you know, they had to work hard to get into the OMOP common data model. To now process all their images is kind of an ask too far. There's a group, there's also a work group interested in imaging, but we haven't really been doing imaging studies. So imaging, genomics, and mobile monitoring are three things we've been doing less of. Again, each of them has a work group associated with it, and some of them have specific use cases. So like on the genomic side, the genomic work group and the cancer work group work together because, you know, they're looking for, there are some uses in cancer therapy where they want to be able to have genomic information, for example.
Okay, yeah. So I was inspired by the principles you talked about sort of verified and open, you know, type of, you know, research and putting out your research questions before conducting the trial to avoid not publishing negative findings. I wonder how that aligns with, as you know, as an investigator, I'm highly dependent on, you know, publishing exciting work. And so that's sort of in contrast to collaborative works in some cases, and also to publishing less exciting findings. Have you had these discussions in your leadership or in the wider team in OHDSI?
So there's a couple things that are possible. First of all, there are a couple journals that are now there for negative results that are trying to, I mean, if you do a carefully executed study that took you a lot of time and shows and doesn't show an effect, that's useful information that would hopefully you would get credit for. And if it's an important enough one, you know, you can sometimes get it published, especially in those journals. The other way is that it doesn't have to be in a journal. It has to be publicly accessible. So once you've done the study, you've spent your time, you might as well get credit for it, partly by putting it. So for randomized trials, we've addressed this through clinicaltrials.org, you know, is one way that you need to register your trial before you're allowed to, has to have been registered before you started the trial in order to publish it in a journal. Yeah. The problem is observational research is being done much more freely, much more voluminously. How can I really track it down? How do I really, like for a randomized trial, there's a lot of work that goes into it and permission. So I can kind of get caught. For an observational study that I can do in a day, how do you know that I didn't just do an observational study, find a good one, then, you know, register it and then rerun it from scratch? You know, so cheating is very easy. So it's hard to solve. I think the way we do it is our process is that we put the results on the internet as part of the process in data.odyssey.org. So the data go there like before we look at the results. So we don't know whether it's positive or negative. But that's, you know, that's Odyssey's, you know, policy. I don't know how to make that pervasive around all of observational research.
Yeah. So George, we're at the hour, and I'd like to ask you the closing question. Where do you see OHDSI and the entire approach, the collaborative sort of data-driven approach in 10 years?
Well, I'm hoping that we can actually, what I see is us creating very useful resources where, because we keep going to large scale. So we should have a website. It's called howoften.org. For every drug, every side effect. Let's just do it. Let's just count it. Like, let's not wait for a case report in the literature to decide we should study and see if this drug causes this. We now have the machinery. Now, it won't be perfect. It won't be as good as doing 1 million randomized trials, but we're not going to do 1 million randomized trials. And if it's reliable enough, I think it's still useful. We're trying to move more into predictive analytics so that we can, like, our big part in that has been reliability of predictive algorithms. So that's where we are, like, make sure it runs across the network, not just in one center, and a number of procedures that ensure that, or not ensure, but push it towards more reliability. But I think being a large-scale producer of evidence that is used by patients and doctors every day is what I would hope would be able to happen. Now, there's limits, just to be honest, like if it's a new drug, there's not in our databases. No. So, we don't see it as the answer in healthcare. It's just a part that we can play.
Sounds really, really good. Thank you so much, George. That was a really fantastic talk. George and I will now move to the neural network. So I invite everyone to stick around and ask questions, and we can chat back and forth. And finally, everyone, please keep in mind the physical summit in Geneva of AI for Good on July 6th and 7th. There's an exciting program being put up. And also stay tuned for more webinars of this health series. The next one will be in March. And again, George, thanks so much. That was really exciting. And I wish everyone a good rest of the day. Bye.
Thank you very much.
Thank you for participating in today's AI for Good session. We hope you've learned something new, innovative, and engaging in today's event. We now encourage you to continue the conversation on the live video wall in the neural network. Here you can ask questions, like and comment, share links, complete the poll, connect with interesting profiles, or speak one-on-one using the chat and video function. We invite you to explore the lobby, try the smart matching quiz, visit the virtual exhibits, poster boards, the eShop, and build your personalized AI for Good program. Let's shape the future of AI for Good.
AI is a powerful tool.
This summit can help ensure that artificial intelligence charts a course that benefits humanity and bolsters our shared values.
you.